This is Dr. Oded Rechavi. He's a professor at Tel Aviv University whose groundbreaking research shows that memories can be passed down over many generations, independent of DNA via small RNA molecules. You heard that right. He and his team have gathered data to not only show that information on past experiences can be encoded in RNA, but that these memories can be passed down over many generations. The reason why this sounds so incredible is because it violates something that we all intuitively understand, and that's the Weismann barrier. But more on that shortly. Dr. Rechavi's work in the small but mighty nematode known as C. elegans is redefining our understanding of inheritance and pushing the limits on long-held dogmas that reach all the way back to Darwin. This work has earned Dr. Rechavi many prestigious awards, as well as being selected as one of the 10 most creative people in Israel under the age of 40. His research is a testament to a fearless approach and commitment to pushing the boundaries of what we consider to be true, something that every scientist should aspire to.
Now, before we get into today's discussion with Dr. Oded Rechavi, it's important to lay down a bit of context. At the heart of our conversation today is the idea of inheritance, but not just in the traditional sense, what we see and understand to be true on a day-to-day basis. We're talking about how traits, behaviors, and even responses to environmental challenges might be passed down to future generations. And not solely through changes in DNA like one might think, but through epigenetic mechanisms in RNA molecules. This idea challenges a long-held belief in biology known as the Weismann barrier, a concept which is central to my discussion with Dr. Rechavi. So what is the Weismann barrier? Well, to put it simply, it's the principle that only genetic information in our germ cells, or sperm and egg cells, can be passed on to offspring. And according to this view, changes that happen to our body cells, somatic cells, like building muscle from working out, suffering from an illness, or even a learned skill like painting that is coded in our neurons and muscle memory, will not be transferred down to future generations, because information is not passed from somatic to germ cells. The Weismann barrier has been a cornerstone of biology since the 19th century, separating what we pass on genetically from the environmental influences we experience throughout our lives, or in other words, the barrier that separates nature from nurture. But in recent years, a growing body of research suggests that the story might not be so straightforward. Enter epigenetics and small RNAs, factors that can influence gene expression without changing the underlying DNA sequence. These mechanisms have shown that experiences can lead to changes in gene regulation that may actually be inherited by the next generation. This is what we call transgenerational inheritance. Dr. Rechavi's research on small RNAs in the model organism C. elegans has uncovered some surprising findings about how memories can be encoded in small RNAs in somatic cells, transferred to germ cells, and finally passed down through multiple generations. Dr. Rechavi's work is transforming how we think about inheritance, suggesting that it might not just be our DNA that defines us, but also the experiences of our ancestors. In today's episode, we'll be exploring questions like how do these small RNAs bypass the Weismann barrier and get transmitted from somatic cells to germ cells? What role does RNA play in gene regulation across generations? And what does this mean for our understanding of evolution, health, and even the choices we make today, knowing they might influence future generations? If all of this sounds a bit technical, don't worry, we'll be breaking it down step by step, and by the end of this episode, you'll have a much better understanding of how the environment might shape not only you, but the generations that follow. So without further ado, join me in vivo with Dr. Rechavi on the genetic transmission of memories.
Dr. Rechavi, I think we should start somewhere with a concept that most people intuitively understand, whether they realize it or not, and that is the Weismann barrier, the so-called second law of biology. What is the Weismann barrier, and how does it relate to how we understand inheritance in the past and currently?
So I think that the non-technical way to think about the Weismann barrier is that our experiences during our life do not affect the next generations biologically. They do not get inherited. If we want to get more technical, then what Weismann talked about is the separation of the soma from the germline. The next generation will arise just from the germline. The germline contains the DNA that and the sperm and the egg, and and the sperm and the egg have their DNA, and after they will fuse, the first cell that will be produced will be the origin of the rest of the cells in the body. What happens to the other cells of our body as parents doesn't have any consequence for the next generations. So the somatic cells, these are the non-germline cells of our body. Whatever happens in the soma stays in the soma. So for example, if I go to the gym and I train and I build up big muscles, these are changes that occur in somatic cells, so they don't affect the next generation. Next generation will have to go to the gym on their own because what happens in the soma doesn't matter to them. If somehow the environment changed the composition of the germ cells and specifically the heritable materials in the germ cells, then the environment could affect the next generation. But that is not something that's supposed to happen, at least when it comes to genetic or DNA-based inheritance.
Right. So, and one of the questions that this is, I believe, like I said, intuitively understood by most people. Like you said, an excellent example, going to the gym, right? Or I guess you can say, the sins of the father or the merits of the father, in this case, shouldn't be passed on beyond the soma. Now, how does this Weismann principle? I get this question often from students. The Weismann barrier. How does it work into Darwin's idea of evolution? We get this basic understanding that we see in biology textbooks that you acquire some sort of beneficial adaptive mutation that allows you to survive, and then it gets passed down to next generations because you have some sort of advantage. But does that mutation have to have existed from day one in the germline?
Yeah. So first of all, when it comes to Darwin, it's important to say that Darwin was probably more Lamarckian than Lamarck.
Yes.
So what we think about as Darwinism today is not exactly as he saw it, at least at some points in his career. But what we think of Darwinism is that the forces driving evolution are variation, selection, and also drift. But we can put drift out of the equation for now. And the environment enforces selection, of course, but it shouldn't change variation. So the idea is that we have different genomes contain different mutations that differentiate us, and these will be selected based on the phenotype that they give rise to by the environment. But the environment itself doesn't change, and certainly doesn't direct the variation.
Right.
And if the Weismann barrier is breached, and what happens in our lifetime changes, especially in the soma, but even if the environment could change directly the composition of the germs of the heritable materials in the germ cells, then that would mean that our life, our experiences affect variation, and would therefore affect the raw materials that selection can work on to bring about the next generations.
Understood. Okay, so we are at a point now where we are starting to develop, specifically you in the space, and others in the space, are starting to develop a sort of second idea in terms of how heritable material can be passed on. At this point, we've discussed the Weismann principle, and we know that the genome is primarily responsible for what we pass down to the next generation. But you have some very interesting evidence, and so do others, when it comes to C. elegans, a wonderful model organism. To put it in context, I believe about 4/5 of the animals on Earth are nematodes, or types of nematodes, C. elegans being one of them. And there's about 57 billion nematodes per human on this planet. So not only is it an incredible model organism, they're everywhere, whether you know it or not, right? As I understand, they're very small, a millimeter or two in length.
C. elegans are 1 millimeter long. You're
okay.
Right.
So why don't you tell us some of the work that you've done in C. elegans and how this is starting to change our idea in terms of models of inheritance?
Sure. So I'll discuss model organisms and C. elegans in particular in a second.
Yes.
But first, with regard to the Weismann barrier and what is possible and impossible in inheritance. So as I said, the DNA shouldn't change because of the environment. The environmental challenges do not sculpt the genome, the DNA-based inheritance. There are when people talk about epigenetics,
yes,
what goes beyond the genome, or I like to think of it as inheritance that occurs independently of changes to the DNA sequence.
Correct.
There are different mechanisms that are studied. For example, there are chemical modifications to the DNA or to the proteins that pack the DNA, the histones. And there are also small RNAs that we'll discuss and elaborate on in a second. Most of the epigenetic modifications or the chemical modifications that occur to the chromatin, the protein and the DNA itself, get erased in the embryo and early development, which means that even if the epigenetic landscape in that sense changes to chemical modifications to the chromatin, even if this landscape is affected by the environment, and it is, there is a reset or a reprogramming that brings us back to a blank page. Exactly.
Yes.
In contrast, small RNAs or these particular types of RNA molecules, they do not appear to be erased or reprogrammed like the other chromatin modifications. And what we found, and also others working in particular with C. elegans, is that in response to different environmental challenges, small RNAs are being made. These small RNAs function by regulating gene activity. Mostly what they do is they downregulate or they silence or interfere with the function of genes. So they turn off gene expression in a very specific way. These RNAs, they match the sequence of particular genes and lead to the silencing of these genes, so they don't get to be expressed and or do not get to produce proteins.
Right, and this is the work that earned Fire and Mello the Nobel Prize in 2006.
Exactly. So Fire and Mello, what they discovered is that double-strand RNA. We all know that the double helix of DNA, that's very famous, but RNA is thought to be mostly single-stranded. However, in certain situations, there are also double-stranded RNAs being made. And what they found is that if you introduce to the worm, this was later found to be true in many, many different organisms, double-strand RNAs, then these double-strand RNAs get chopped to produce small RNAs, which then lead to silencing of different genes. If they find a gene that matches them in sequence, they lead to its degradation using many proteins that help them to do this. And what we found, and this is now a field that many work on, so when I say we, I talk about the scientific community, is that in C. elegans, in these tiny nematodes, different environmental conditions lead to the production of small RNAs, and small RNAs that not only act in the parents, but also get transferred to the next generations and lead to silencing, so they alter gene expression, not only in the progeny, but in many generations down the road. And this changes the physiology of the next generations in response to what happens to the parents.
Right. Okay. And, we're going to use the term memory from time to time. And it's important to understand that, at least as far as the space is right now, when we refer to memory, we're referring to memory of challenges that have been pushed or placed upon whatever organism you're using, in this case, C. elegans. So this might in the future extend to actual coding of memory in neurons being reflected in some sort of RNA, rather. But we're not exactly at that moment right now. So just for context, the audience should keep in mind that memories mean some sort of challenge and a response.
Or memory, in my book, means that something is different because of what happened in the past.
Yes, and has been recorded somewhere, captured somewhere, the information of such. Right.
Mm-hmm.
Okay. Now, when you something that occurred to me when I was researching your work and when I heard it for the first time, you're referring to the transfer of these RNA molecules, which I wanted to mention on just as a side note, is now a widely used technique, thanks to Fire and Mello, in any sort of molecular genetics lab or molecular biology lab. If you want to silence a gene in some tissue culture or in an organism, you use this technique of double-stranded RNA that's complementary to the gene you want to knock down. So thank you, Fire and Mello.
And there are even drugs.
Yes, that's right, that have been approved. Yes.
Mm-hmm. Mm-hmm.
Okay, so what occurred to me, and I love to approach molecular biology from base principles thinking. And when you mention the transfer of RNA, are you referring to literal transfer of RNA molecules into the germ line or the germ cells of the parent that then survive fertilization? In this case, in nematodes, they're hermaphrodites oftentimes. But it's the literal transfer of the molecule, or is it the transfer of the non-coding region in the genome that codes for these RNAs that's being transferred?
Okay, so there's a lot to unpack here.
Yes.
Okay, First of all, in C. elegans, we know that there is systemic transfer of RNA between cells and tissues.
Okay.
And this is less understood in other organisms, although it does occur in many different animals and certainly in plants. In C. elegans, we know quite a lot about it, but not everything that needs to be known. For example, there's one gene, it's called SID1, that stands for Systemic Interference Deficient 1, which codes for a channel that helps double-strand RNA move between cells. And indeed, if you introduce double-strand RNA to one tissue in the worm, it leads to silencing not only within the particular tissue that you injected the RNA into, but also in the rest of the body, including in the germ cells. And this is very clear, and there are mutants where this doesn't happen, defective in these C genes.
Understood.
So RNA can move from the soma to the germline in C. elegans. Some of this process, it's unclear whether all of its process, but some of this process is certainly mediated by this gene SID1, and there are other genes that affect it. Once the RNA reaches the germline, it can lead to silencing there, but in order to survive for additional generations, it needs to be amplified. What do I mean by amplified? Mm-hmm. So every worm produces hundreds of babies, about 250 on average, something like that. Mm. So after a few generations, and most heritable small RNA-based gene regulatory responses last for multiple generations, so 3, 5, or even more, in certain cases, hundreds of generations. After a few generations pass, the dilution factor, if it was just this RNA persisting, would be enormous. So after 4 generations, it would be 250 times, 250 times, 250 times, that would be 4 billion or something. Totally homeopathic, would never work.
Right, completely diluted, doesn't exist anymore, essentially.
And people have shown that some RNAs, there was a recent paper in Nature or Science, I don't remember, about a long non-coding RNA in mice or rats that persists for years. So this is just this one RNA persisting for a very long time. And this, in fact, I think was shown even before in certain organisms. But in the case of C. elegans, this is not what's happening. What happens is that the RNAs get amplified over and over and again by these enzymes, complexes that are called RNA-dependent RNA polymerases. Mm-hmm. What they do is they use RNA, the gene's RNA, the messenger RNA that the gene encodes for, as a template for producing more and more small RNAs. So once you have one small RNA, it can bind to this mRNA target, and it recruits the RNA-dependent RNA polymerases to the mRNA target, which then uses it as a template to synthesize, to polymerize more and more and more small RNAs. And this is a sort of a feedback that then leads to the production of more and more small RNAs. In fact, the process is much more complicated. We don't fully understand, but we know, for example, that it entails chopping of the mRNA and tailing of the mRNA with untemplated nucleotides that gets added to the mRNA, which then serve as a sort of a landing pod for the recruitment of another round of RNA-dependent RNA polymerase that will make small RNAs. So it's more complicated. Well, when I talk about RNA memory, I mean the RNAs get passed to the next generations and lead to the production of more small RNAs, and this is how the cycle continues.
Right. So essentially you can summarize it as saying there is de novo RNA synthesis occurring at some point.
Absolutely, in every generation.
And so the transfer of the RNA through these channels into the germline, do you imagine this happening like a passive diffusion, or is there evidence that it is somehow regulated or controlled?
To tell you the truth, we don't know enough about it. Okay. Most of the evidence, I mean, we know that it spreads. That is obvious from many experiments, including the original Fire and Mello paper. They already showed it there.
Yes.
And we have mutants where this doesn't happen. Exactly how this happens, it's unclear. In the beginning, for example, SID1, people thought that this is a channel that sits on the plasma membrane, and actually the RNA gets, passed through. Now we think that these channels actually sit on vesicles inside the cell and help release the RNA from the vesicles to the cytoplasm. How is it regulated? How dynamic the process is? Is it gated? Is there selection of particular RNAs? And if so, which RNAs? We don't know.
Before I delve into some more specifics, why don't you give us an example from your work with C. elegans and the sort of memory that is being transferred, that some of the challenges with your work with C. elegans and viruses and some of the other experiments that you've run?
Yeah, so for example, one of the most interesting papers we've been involved in is we've shown that RNAs, endogenous RNA molecules that are made in the nematode's brain or nervous system, affect the behavior of the next generations. What we did in this paper, this is the Posner et al. Paper in Cell 2019, is we took worms that are defective in a gene that's called RD4, which is required for making small RNAs. It's required for making certain endogenous small RNAs and also for functional exogenous RNA.
Is that an RNA-dependent polymerase?
No, this is a double-strand RNA binding protein.
Okay.
It acts relatively upstream in the process.
Okay.
And so we took these mutants, and we found that they don't make certain small RNAs. This is clear. There are genes that are misregulated in these worms, and we also found that in relatively high temperature, in slightly stressful temperatures, 25 degrees Celsius instead of 20, these worms are defective in finding food, in chemotaxis. They don't behave properly. What we found is that if you take the missing gene, RD4, and you express it specifically just in the brain of these worms by driving its expression using neuronal promoters, it rescues the capacity of the worms to properly find food and behave. Mm-hmm. But more interesting, and also it fixes some of their problem in expressing specific genes, which we monitored using sequencing and also different reporters. But more interestingly, also the next generations of these worms, even if we lose this gene that we rescued in the brain, so they are back to being mutants.
Right.
The next generations behave fine, and also they properly regulate the genes that depend on RD4, which means that the production of small RNAs in the brain of the parent was enough to correct gene expression and also behavior, in this case, in the next generations. And this is what we know. We know that this heritable epigenetic effect depends on factors that are required for RNAi, for small RNA induced gene regulation in the germline. Right? There's a gene that's called HRD1. This stands for heritable RNA deficient 1. If you don't have this gene, this is a germline specific gene, and it is needed only for heritable small RNA regulation.
That's its only purpose.
Yes, I mean the parents are okay with doing with silencing genes, but it's not inherited.
Interesting.
Right?
Interesting.
And this gene is required for this process of transferring the brain effect to the next generation.
Okay.
However, this particular effect that we found was not dependent on SID1. Or it was, to be more accurate, it was partly only very loosely or weakly dependent on SID1. And this does not mean that SID1 is not the only channel that transfers RNAs from the soma to the germline. But it's possible that when we disrupted, and this is where things get complicated, and I have to admit we don't understand what's going on. Mm-hmm. When we don't make particular small RNAs, this then changes the balance of all small RNA making in the cell. There are many different types of small RNAs. We didn't get into it yet, but many of them share a common biosynthesis factors, which they compete over. And these are limited resources. Mm-hmm. So when you affect the production of one small RNA species, indirectly you also affect the production of another small RNA species. So when you don't make RD4-dependent small RNAs, it is possible that you affect the production of other RNAs, which then don't move properly from the soma to the germline, leading to this sort of a snowball effect.
Mm-hmm. Understood. That is fascinating. That there's almost a way to recover the mutations and the controls that you're running. You're knocking down in the parents, and there's a way for the second generation not only to inherit the memories, the RNA, but they're also tinkering with your experiments in the sense that you mutate the parent and they have a way to recover. It's fascinating.
It's fascinating. It's fascinating. It's also complicating a lot of things. So normally when you think about, I have a mutant.
Right.
That's phenotype.
Right.
But you have to take into account all the non-DNA based inheritance and everything that happened in the past. So yes. When it comes to C. elegans, this affects very practical things like, for how many generations were the worms starved? Or
right.
well, did I always grow them for multiple consecutive generations in a particular temperature? If not, then this could affect the phenotypes of the next generation, even if you think that you know what is the genotype that you are studying.
the the first thing that comes to mind when you say that is you're introducing, in the normal sense, in the wild type sense, when you're running experiments, the the uh, the possibilities are almost endless. And I think of a reviewer on a paper, for a long time, it's
I'm actually dealing with this
really.
hypothetical viewer. No, I mean always.
Right, right. Well, in molecular biology, when you're doing transfections, and the review is always off-target effects. Have you considered this? When you put this into the equation, it's almost you can't control for anything at some point.
Yeah, so we were thinking about, for example, ways of resetting this inheritance.
Mm-hmm.
One thing that we played with, and we also published this, is this inducible degradation of the gene that I mentioned before, HRD1, which is needed just for small RNA-based inheritance.
Okay.
So you remove this gene temporarily,
Yes.
then you bring it back, and so you sort of reset RNA inheritance. And but that's also not the ultimate solution, because, as I said, when you don't make one type of small RNAs, you make others as a result. So you don't really know. It's as if you can't put your feet in the same, how to say, dip your toe in the same river twice. I don't remember how.
No, you've got it. You've got it. That's true. Yes. Okay. So how does this in ability to inherit RNA affect whether or not generations of C. elegans can be infected by viruses? How does it affect in a sense, their immunity or their immune system, which is, for lack of a better term?
Right. So remarkably, worms are almost completely resistant to all the viruses that we know. So there are very few viruses that infect C. elegans.
Mm-hmm.
In fact, there's only one virus that infects C. elegans that we know, which is called the Orsay virus. It was identified in a rotten apple in France in 2011, I think, or 10, I think 11. No, I'm not sure. And they found this worm in the apple which had the virus. However, they later found that this worm is an RNA mutant. The same virus, if you try to infect wild type worms with the virus, they are very good at dealing with it. And we found that another nodavirus from the same family, which is artificial, if you infect the parents, it leads to the production of small RNAs that then prepare the next generations for dealing with this virus. And Craig Mello, a few years later, showed that this happens with another virus. Mm-hmm. We think that RNAi, I keep mentioning this, I'm not sure whether we define it, is RNA interference. This is the process that the small RNAs use to silence genes. So RNAi probably evolved as a way of dealing with parasitic genetic elements, mm-hmm. viruses and transposons, jumping genes, repetitive genes, and genes that don't function well or to your advantage. And so, and indeed, we find in many different organisms that small RNAs regulate such genomic parasites, transposons and others. And it's a sort of an arms race between small RNAs and the elements, the genetic elements that they regulate, and they keep evolving, avoiding each other, and then finding each other again. And this is probably the origin of RNAi and why it exists.
Interesting.
And the worms are very good at dealing with viruses using RNAi, although they don't have T cells, B cells, antibodies. Just using RNAi, they do this very, very effectively.
Now, in terms of virus infection in C. elegans and the RNAi, so it's the RNAi that they are, or rather the RNA that they are producing that inhibit the viral genes and degrade them to prevent infection. That RNA that they are transcribing is encoded in the genome of C. elegans, right? I guess what I'm trying to draw is a parallel between the way that C. elegans produce RNA and humans produce antibodies.
So it's not exactly the same. So there are endogenous RNAs that are made in the genome.
Yes.
Regulating genes transgenerationally. But in addition, when it comes to the virus, it's the fact that the virus, when the virus replicates, this particular virus that we're talking about, or this virus family that we're talking about, during the course of the replication of the viruses, the virus makes double-strand RNA. And the double-strand RNA is then the signal. The cell looks at the cell and says, what is this double-strand RNA? This is suspicious. Maybe it's a virus replicating. is why the double-stranded RNA get chopped, small RNAs are made, and the small RNAs are then go and attack the virus again.
I understood.
Okay, but when it comes to other small RNAs that get inherited, some of them derive from endogenous double-strand RNA. There are double-strand RNA molecules also encoded by our genome. And some small RNAs are not made from double-strand RNA. They are made there are single strand. The origin is a single strand. I would also say that in many cases, it's some weird feature of the target that triggers small RNA production and then transgenerational silencing. When it comes to the virus, we just said this is the fact that it makes double-strand RNA, but it could be other things. For example, there are hotspots in genomes, and this was mostly studied actually in flies, not in C. elegans, which make small RNAs that then silence the gene that they correspond to transgenerationally. Okay? These small RNAs are called piRNAs, and they are a little different in C. elegans and in other organisms, but we don't need to get into that. So there are hotspots in the genomes which make these small RNAs, and if you get there, if you're a gene and you landed in this spot, you will be targeted by small RNAs because the small RNAs are made there, and you will be silenced. Okay? So you can think of these genes, areas in the genome as traps, genomic traps. Mm-hmm. Why would you land in such a trap? Perhaps because you are a jumping gene, a transposon, which is a foreign parasitic element. So this uses the one thing you can say about transposons is that they jump. And if they happen to jump in the wrong spot in this trap, then they will be targeted by small RNAs and will be silenced transgenerationally. So it was a very smart or elegant way of the cell to use the weakness or the only thing that transposons need to do to jump against them. Mm-hmm. And there are other marks or other features of genes that lead to their silencing. For example, if a gene is very weird, it doesn't have introns, let's say, or it has a totally different codon usage, and it will be recognized as foreign, and the RNAi system will recognize it and silence it. Okay? Some people even suggest that there's a beautiful review paper, hypothesis paper by Heath and Medani. It's called something like the frustrated gene or something like that. And the idea there is that transcription, the process of transcription is so elaborate and complicated to recognize foreigners or things that the gene wants to silence that are not of its own, that not self, that non-self. Okay? So it's like you go to a dinner with the Queen of England, and you have many, different types of forks and knives. And if you didn't grow up in the royal family, you don't know how to use them. So it's the same thing. If you're a new gene, a virus or a transposon that landed in your genome, then your transcription won't be exactly the same as the host.
Right.
And the fact that you don't know exactly how to behave will be used against you to silence you. It's a way to recognize that you need to be eliminated.
Interesting. Definitely not the same as our immune system, but there are some interesting parallels there. Just the fact you self non-self is something that we use in immunology all the time.
Right.
So for how many generations have you shown or have others shown that this sort of memory or genetic or inheritance of RNA can persist for? How far?
So typically in many experiments, we and many others have shown that the typical response at the population level would last for a few generations, normally 3 to 5 generations. However, in many responses, some individuals would continue to silence for a lot longer. And in certain situations, the entire population will continue to silence for a lot longer. And there are responses that last for hundreds of generations or many years. C. elegans generation is very short, it's just 3 days.
Right.
But there are some responses documented that last for hundreds of generations, 5 years, so on.
So I know we're getting into some speculation, and I really like that and appreciate that about you, because you're willing to speculate from time to time. It makes things interesting. Do you think that that's we spoke about dilution over generations, 250 times 250 times 250, for example, but why might it be lost? Does it come to a point where that the ability to amplify that RNA, I mean, obviously that ability is getting lost. What does that do you think?
Right. So again, as you said, rightfully, I'm speculating. Okay, we know that there are mutants where this lasts longer.
Mm-hmm.
Okay? So we know also that repeating an environmental challenge would extend the duration of the heritable response. So let's say if you put the worms in high temperature, you'll get and then you bring them to the normal temperature. It will last for a few generations. But if you grow them for a few consecutive generations in high temperatures, the effect would last much longer. So it can be boosted. Okay, and the question is, why does it stop, or why does it continue? And there we don't know. If we're talking about the adaptive value of it, we have no idea. And in fact, it could be, so if we're talking just about the mechanism of it getting lost, it could be because you have some effects stemming from the outside. Let's say the production of a small RNAs against a virus, and then the virus is no longer there. There's no more template, and the small RNAs that are made against the virus are competing over with small RNAs, endogenous small RNAs that are made using the same machinery, and they just chelate them out eventually. That's one possibility. Okay,
Right.
But if we're talking about adaptive value, or why is it timed to last for a certain number of generations, then we can only speculate. We could say, for example, that the response to a temperature would be correlated, let's say, with the seasons.
Mm-hmm.
It would last throughout the course of a season. But that's pure speculation. I have no idea that this is the case, okay? I'm just thinking, giving an example. And the fact is that we just don't know enough about the worm's ecology. There are great models for many, many, many different things, but we're only starting to understand their ecology and the natural things that they encounter and have to deal with. And maybe this could be a good clue for why the epigenetic responses last for as long as they do.
Right. I would imagine that along the lines that you're talking about. I mean, biology has a remarkable way of finding the path of least resistance most of the time. And so if some sort of pressure no longer exists, why divert resources to making that sort of RNA, for example? I think essentially is what you're describing there.
Right.
Now, one of the reasons why I asked this question is because one of the standard descriptions of RNA in biology is that they do not last for a long time because they have an extra oxygen, ribonucleic acid. There's an extra oxygen as compared to deoxyribonucleic acid. So they're prone to attack. They're often single-stranded, which is prone to attack. How is it that these RNAs can sort of evade that basic paradigm that we teach students so often about RNA?
So I think that, first of all, as we said, there are examples of RNAs being very long-lasting, like these long non-coding RNAs that I described before. Mm-hmm. And plus, the individual molecule might not survive, but it is being amplified and remade.
Right.
And probably RNAs are never alone in the cell. These small RNAs are always inside some protein. There are proteins that are the effectors of small RNAs. They are called argonauts, and they are probably always carried by some protein that protects them. But regarding this dogma of RNA being super, super sensitive, in C. elegans, we can just feed them on double-strand RNA. We can soak them in double-strand RNA, and it's taken up and it's still effective. Perhaps when they're small, they're less sensitive.
Understood. Now, how do you imagine that this transfer, now to lay down some sort of context, nematodes, or C. elegans in this case, sometimes act as hermaphrodites, and sometimes they mate in a classical sense. But how do you imagine that this transfer of RNA differs between how it gets into sperm and how it gets into egg? Because we know that at least in mammals, females are born with their full complement of eggs, and men are constantly making more sperm. So how do you think that changes the ability to inherit? And how do you envision that the transfer is different, or is it the same?
Well, so one thing we know first is that RNA inheritance works both through the egg and through the sperm.
Okay.
In the past, I mean, as you said, C. elegans are hermaphrodites. They make both sperm and egg, but there are also males in the population, which are relatively rare. But we know that both females, if we want to refer to the hermaphrodites as females, and the males can both transfer RNA responses transgenerationally. There are factors that are unique to the sperm or to the egg that function in small RNA biology, and they are unique. For example, different argonauts in the sperm and egg that carry a different complement of small RNAs. Exactly how the small RNAs get selected to the many argonaut proteins that the worm has is unclear. And if we want to speculate again and talk about the reason why there might be differences in inheritance via the sperm or the egg, then often in evolution you think of males as sort of agents of chaos,
Yes.
introducing variability, and this is certainly true when it comes to C. elegans. And you have, since the hermaphrodite can self-fertilize, they can make something which is not a perfect clone, but it becomes more and more, but it's almost genetically identical. When you mate with a male, you introduce increased genetic variability. You now mix two different genomes, and these males might also transmit RNAs that would again increase variability. I would also add to that more males are being made under stressful conditions. So normally in a plate of C. elegans in the lab, males are only 1 to 1,000, but in the wild, they are much more common.
Okay. Is that because they're more difficult to culture?
No, this is just because this is the rates of their production.
I see.
Mhm.
I see, understood. Okay. A question that has stood out in my mind is how much time must elapse before some sort of stress will have the ability to affect the next generation? Can you stress them? For example, you speak of temperature, high heat, low heat, or viral infection. Can you stress them and then immediately have them mate and have the next generation? Or does this stressor have to have time to sort of build up more RNA? How do you speculate the mechanism works there?
Yeah, so
With the constraints?
us C. elegans researchers, we're a little spoiled. So when I talk about experiments and I'm saying, so transgenerational experiments are long because they take multiple generations, it's true that it's not mice and every generation is short, but still it takes a while.
Yes.
So if we do this type of experiments, let's say put them in a high temperature and I'm examining the next generation, would take me a month or so. So I admit that we didn't do many games with trying to change, to put them in for 1 hour, 2 hours, 5 hours and see how this changes. So I really can't answer. What I can tell you are the specific, so we didn't do enough, okay? So I don't know. But we did show previously, we have a paper for, I think, 2021, but I'm not sure, Huri et al. At eLife, where we showed that both, so they're putting the worms in high temperatures for their entire life or for just 2 hours generates a transgenerational effect. Okay, exactly how the effect differs or whether a 1 hour effect would also lead to something, we don't know. But it's just, there's so many iterations, so many variations you can do, and it takes we need the entire community to do this.
Right, and interest is obviously growing thanks in part to your lab. Now, are there any examples in more complex animals than C. elegans? Not that they're not complex, but something more complex that might be sparking interest?
Yeah, so there are many examples of transgenerational effects in many organisms, in many animals, certainly. I mean, in plants, there are very well-established experiments. In other animals, especially mammals, it is still very controversial. And sure, there are examples of this, of all kinds of parental effects transmitting for multiple generations, but since we don't understand the mechanism, they remain controversial.
Right.
But you have endless examples and people summarize it in reviews, but it's still, I admit, very, very controversial. What our angle to this is we try to work with animals where we can do very well-controlled experiments and understand what we're seeing. And I can't elaborate too much on the details because this is not published yet, but we now have example also for transgenerational RNA inheritance in flatworms, which are also worms, but totally different, separated by hundreds of millions of years.
This is planaria?
In planaria, exactly. And there it's very obvious, and as I said, we didn't publish it yet, but the effects are very clear, that there are very, very long-lasting effects of RNAi, probably by mechanisms that are a little different from what's happening in C. elegans, but the end result is the same.
So blink twice if it's similar to McConnell's work.
No, so it's not similar to McConnell.
Okay.
I can talk about that if you want. So McConnell was a psychologist working in the 70s and 80s, suggesting that memory, but real memory, memory that is encoded in the brain, transfers from after you chop off their heads and they grow a new head, and also when you chop them up in a blender and feed them to other worms. And this is extremely controversial, it's a whole story. We didn't work on that. We work on actual RNAi responsive worm. We feed the worms with double-strand RNA and look at the sciencing of a particular target.
Now, you've mentioned now a couple of times in the last couple of minutes the term controversial, and that's because the history involved with the idea of inheritable traits goes back to and originates with Lamarck, and there's a bit of a stain on this research because of some controversies. We don't need to get into the controversies; they are interesting, and I encourage people to look them up. But my question to you is, even this work today is controversial. How do you allow that controversy to impact you or affect the way you do your work? How do you approach the way what you do in the context of that it might be considered controversial? Does it affect you?
Yeah, so I think first of all that inheritance of acquired traits, slightly different from transgenerational epigenetic inheritance, but certainly connected. It's probably the most controversial topic in biology, in history, hundreds of years. And this is because of many reasons. Some of them are these controversial episodes like McConnell, like others, and some tragedies like the starvation in the Soviet Union, the Lysenko affair. So different clouds on this entire field. And some because it messes up biology. It's complicating everything.
Right.
We think we figured out everything with genetics, and then, as we said, when we discussed about all the controls you need to think about, it just makes things complicated.
That's right.
And there weren't any good mechanisms to explain how it might happen, so it remained controversial. When I started working on this in Elegans, it was very controversial. Now I think that the situation is that everyone pretty much agrees that it happens in Elegans. So I think in C. elegans, it's not controversial anymore. In other organisms, it is controversial. It's not controversial in C. elegans because it's so easy to do. You can just feed them with double-strand RNA, get sciencing translation. But in any case, I have no, and I'm a little disappointed that it's not controversial anymore, but it makes it more interesting. Okay. Once so I don't want anyone to think that this is solved. It's not solved. There are many things we don't know. As also, we talked about some of the unknowns, but not about all of them. But I think that it remains controversial in other organisms, and I'm totally fine with it. It means that it is interesting that we are at the boundary, at the frontier, and I think this is where we should look for scientific questions.
So yeah, absolutely. And what I think I hear you saying is that you're not afraid of controversy, and that in fact it helps you thrive in this space. And I think it does. The fact that it is slightly controversial means that it gets exposure, and that's how I came across your work. And we're sitting here talking about it today, and more people are going to hear about your work, and that's a great thing. Now, one of the things that I really love about this sort of paradigm, this concept of RNA being transferred, is the fact that it diverts your attention away from DNA for a second, because of course it's always been considered the most important molecule. What it gets me to think about is the fractal nature of information that's stored in any given cell or any given organism. It's enormous and it's fascinating. You have DNA that codes for RNA, that codes for protein in an amino acid sequence, which can then itself go back and modulate DNA and introduce epigenetic modifications. It's almost endless. So do you envision, what do you think about this idea of the fractal nature of DNA? And do you envision a time where we will completely understand the breadth of the way that information is stored in a cell?
I think what it means is that it is very dynamic.
Yes.
It is very dynamic. We don't, unless we record all of the molecules in a cell, we won't have the full picture. So that is clear when it comes to DNA. We know that, it's not enough. You just put a DNA in a new cell, it won't be exactly the same. There will also be the history, the cytoplasmic history, and everything that it carries. And I think that when it comes to the fractal nature, I'm not sure that the picture is always the same in every resolution that you look at.
Oh, certainly not. Yeah.
So that is an interesting thought.
Now, if we may, with the time that we have left, speculate a little bit and move towards sort of the ephemeral. Um, how do you envision how this might work in humans? To what extent, if it does, the transfer of RNA? We know that it does a little bit. To what effect we don't know. But how do you envision where this might play out in, say, the next 10 years?
Mhm. So it's very hard to study in humans.
Yes.
There are people who say it can't happen in humans. Sometimes use um, this sort of a logic. They say, no, in worms it makes sense. As I said, it's not controversial anymore with Elegans. They say in Elegans it makes sense because the generation time is so short, it's just 3 days. So it makes sense to prepare the next generations for what the parents experienced.
Mm-hmm.
The likelihood that the environment that the parents live in would be similar to the environment of the progeny is high. So it makes sense to prepare them for
Right.
what you expect. Okay,
Mm-hmm.
and also they don't move so far because they're small. So
Mhm.
maybe environment say my kids, might live in a different country. Who knows? It will take 20 years. But I think that this is not necessarily, a great explanation or a reason, because there are different scales to things. Like if you're talking about viruses, we encounter the same viruses for many generations, or similar
Right.
families of viruses. So it doesn't have to be a real reason for it not to occur. It's very hard to study in humans because we can do it's hard to do experiments. We don't want to do experiments on humans,
Mm-hmm.
and the generation time is so long, and we are very different genetically. So the worms that we study in the lab are almost identical in terms of their DNA, and also they live in a very defined condition. So it's easy to separate nature from nurture. In humans, that's hard to do. Another reason why people say it can't happen in humans is that in C. elegans, it really depends on these RNA-dependent RNA polymerases that amplify the small RNAs.
Yes.
And we humans don't have them.
Right.
However, there are other mechanisms that humans have that C. elegans don't have, like DNA methylation.
Right.
And also, I just, gave you a scoop that it does happen in RNA in planaria, and planaria don't have RNA-dependent RNA polymerases as well. Okay? So there could be different tricks. There are all kinds of feed-forward mechanisms that might maintain gene regulatory responses. So I really can't say anything about humans aside from really speculations. I think that we will get there someday to understand. And probably the key is establishing a very good system, because even the experimental design of such an experiment is not clear. It's very
Yes.
challenging.
Yeah. The design itself you have to overcome before you can even set
Right.
forth, right? Now, how do you envision the work that you're doing and similar groups are doing becoming actionable in the future? Do you think that this will manifest in some sort of um, health outcomes, pharmaceutical? Definitely, I can see, and you've probably thought about this, research techniques and tools like RNAi constantly evolving and improving. So I see that absolutely. But do you have an ultimate goal in terms of actionable consequences?
So first of all, we need to. I'm more interested in the basic question first. But
Yeah.
let's say it does happen in humans. It does happen in humans, and the full disclosure, we have no idea that it does.
Right.
As you said, there are 4 out of 5 animals on this planet is a worm. So even if it's just in worms, it could be very important. But
Yes.
let's say it does happen for the environment. But let's say it does happen in humans. Then the first thing I could imagine is that we would um, um, monitor the pool of small RNAs. So we will sequence the pool of small RNAs in sperm and try to correlate that with some health outcome. Just like any couple bringing a baby today, they do some genetic testing looking at the DNA, but it could be that there's a whole additional layer of the RNA that is small RNAs that are in the sperm and the egg. The egg is, of course, less accessible, but sperm is accessible, that you can try to sequence and then correlate it with different traits and find that so long before you act on it and introduce an RNA or remove an RNA, you could first inspect it like you do with DNA,
Right.
and try to find correlations to different things. And I think there might be something there. It's a huge endeavor to set up something like that, but it's possible.
Now, if I may quote you from your TED Talk, you said, if memories are inherited in humans, it might mean you have a greater responsibility for your actions. But regardless, it's probably a good idea to act as if it does. I think that's a fantastic outlook, a philosophical approach to what you're doing.
Thank you very much. Yeah, I think it's better to act, consciously and be responsible and think
Yeah,
about the
absolutely.
next generation as if you are affecting them, even if you are not.
Yeah, and the sort of idea that probably would only cross the mind of a molecular biologist, and that I can appreciate. Something else I wanted to just move away from this work for a second. Another reason why or how I came across you is your obvious ability and skill as a scientific communicator, and you have a very unique way of doing that today on X, and that is through the meme. And you are probably my favorite science account on X, and your memes are powerful. Your meme game is through the roof. There are some people who can meme, and you can. How did you come to this? Did you read PhD comics when you were doing your PhD? I don't know if that made its way to your side of the world.
I did, and I loved it. It's
Yeah.
great. I can't say that it affected me or that I tried to, do the same. Probably not, but I certainly loved it, and I think they're great. No, it was totally by accident. I joined Twitter on 2016. We just published a paper. I wanted to, let the world know, so I tweeted it. There was barely any response, although it was a Cell paper, but no one cared. And that was probably. And so so I said to my friends, I don't understand this social media. I don't know how to do it, but. Then I don't know how even I started making these stupid jokes about science, and it resonated. And I think that this is because there's a lot to joke about. The science is serious, but it's also fun. And the academia and this weird world that we live in that will be very weird for outsiders. They won't understand what's going on there. And some people, non-scientists, tell me, your Twitter account is so niche, we don't understand what's going on there. But I think scientists, they do understand, and it's a twisted world. So there are many things to joke about, and people connect that because it really affects our day-to-day life and our also long-term plans that we have to deal with this weirdness.
Yeah, science is definitely serious, but there is also a large need for some levity that you bring, right? And also to communicate with other scientists that, hey, we're out there and we're in it together and we understand each other, right? Okay, just a couple of last words, if you don't mind. Words of wisdom for young scientists. You've come a long way in your career. You're a very successful scientist. What does it take to be a successful biotechnologist, molecular biologist, geneticist in today's world? What are the skills that you look for when you hire in your lab? And any words of wisdom and advice for people coming up through the system now?
So I think that the system encourages you to be a coward. I think that the way peer review works and committees and PhD committees, and it all encourages you not to take risks and to be on the safe side. And that's very so. And you need to be the one criteria that will really determine whether you'll continue in the business or not is if you enjoy it. I mean, people quit science because they don't enjoy it. It's not because they didn't publish in Nature. It's because they stopped enjoying it.
Right.
And if you just stay and do safe things all the time and just try to do the same because that's the path of least resistance, like you said, and will always be afraid of your own shadow, then at one point you will stop enjoying it. And the temptations from other places, from the industry, from high tech, there are many temptations, many places where you can make more money and it's easier. But there are great advantages to being an academic. The freedom that you have, the creativity, that it doesn't need to end up it doesn't have to be useful. It doesn't have to need to make a profit. There's so many good things about science, and it's a great way to live your life, but you have to enjoy it. So make sure that you take care of enjoying it first.
Yeah, I think one thing that stood out to me when you said that is the freedom. Oftentimes students will ask me, when you were doing your PhD or PhD master students, how do you decide day to day what it is that you do when you arrive at the lab? And really the answer is whatever you want. I mean, you're being directed by the PI most of the time or some sort of mentor, but whatever the lab wants to do. And that freedom is incredible
Right.
and
People don't
necessary.
understand how unique this is.
Right. Exactly. Oh, well, Dr. Rechavi, it was an absolute pleasure talking to you. I look forward to the planaria paper. I'm sure you'll blast it out on X when it comes out, and we'll make sure that your X page is linked in the show notes below. It was an absolute pleasure, and I hope we can do it again sometime in the future.
Thank you very much. Totally my pleasure. Really enjoyed it. Thank you.
Have a great day.
You too.